Metabolism, as all of you know, has two parts, anabolism and catabolism. Anabolism deals with the synthesis of molecules. Catabolism is about degradation of macromolecules into polymers into monomers, let us say. These two together we call it as metabolism. Of course, for metabolism basically is about utilizing your food and creating energy from it to do whatever work the cells have to do. Basic thing is cancer cells also need energy to proliferate. Ultimately, they have to proliferate. So, where the energy comes from, which pathway they utilize. So, that is what is the important question. If you remember, I told you about the 6 hallmarks of cancer in one of the introductory classes, and I also told you there are some emerging hallmarks. So, one of the emerging hallmarks is the deregulation of cellular energetics. Of course, avoiding immune destruction also we have talked about it. The enabling characteristics are genome instability and mutation and tumor promoting inflammation. So, these are the important or the so called emerging hallmarks as they say. So, deregulated metabolism is also a hallmark of cancer. So, definitely in cancer there is some metabolic deregulation. Let us look at some of the historical features. It was in 1887 that Freud proposed that cancer cells can be killed by reducing glucose. That is, he actually advanced a theory that said the blood of patients suffering from cancer contains an abnormal quantity of sugar and that cancerous growth be destroyed by a reduction of the amount of sugar. That is what he kind of said that. Does not mean that all cancer patients are diabetic, it is not like that, but he said that there is some need for more amount of glucose. In that sense, he is okay. Muller observed somewhere in 1890 aberrant metabolism in the urine of cancer patients. So, basically people started observing some problem with metabolism in cancer patients. So, that is how it all started. In 1912, Wasserman hypothesized a role for deregulated respiration in cancer cells. So, he thought that, see, it is nearly 100 years ago. Notice that cancer cells are growing rapidly, their oxygen requirements would be different from greater than those of the normal cells. So, that is what he postulated. But by far the most important thing that has been revisited now is what is called Warburg effect. Warburg, as the name goes, Otto Warburg, he noticed an increased glycolysis in cancer cells somewhere in 1924. This is a very important observation. His main contribution is, all of you must have studied glycolysis and TCA cycle, where there is this oxidative phosphorylation is involved in the TCA cycle, and also remember about the locations. Glycolysis takes place in the cytosol, whereas TCA cycle in the mitochondria. So, the cellular compartment is important, and Warburg observed this increased glycolysis, which means he said this oxidative phosphorylation is less, whereas that is it does not go to the TCA cycle, but instead produces something like lactate, which is usually produced in normal cells. It is done under anaerobic conditions or when you are doing some muscular work, something like that. Here, even in the presence of oxygen, you get lactate. So, it is aerobic glycolysis only, even in the presence of oxygen it goes to. So, this is in a way, as we go on, I will tell you the number of ATP molecules produced by our regular oxidative phosphorylation for one molecule of glucose will be more than 30, whereas with this anaerobic glycolysis it will be only 2 ATPs. So, it is a very inefficient process, but even then the cancer cells appear to prefer this, and so if you have to have more energy, then that means more glucose is needed. That is the point. That is, the cancer cells need more glucose and they need more energy. So, this is what is happening, but this happens even in the presence of oxygen is the important point. Normal cells, this anaerobic glycolysis takes place. So, that is what he kind of proposed. But there was some debate. Warburg published on the origin of cancer cell somewhere in 1956 also, and there was a debate between other scientists like Winehouse on mitochondria and cancer, because Warburg was telling there is some problem with the respiration. He said that the respiration won't be proper, but that is not actually true. The respiratory properties are fine even in cancer cells. So, that created some rift between different scientists. And then it is Kovacevic who observed somewhere in 1972, after all, if you are not utilizing TCA cycle, it is not that they are not utilizing, they are less utilizing relatively. That is how you have to take it. It is not that there is no oxidative phosphorylation at all in cancer cells, that is not the correct thing, but it is less utilized. So, if it is happening that way, the other requirements of the cells will be affected. So, that is taken care by glutamine. Glutamine also can be utilized. All of you who have studied metabolism should know that energy can be derived, of course, mainly from glucose, but also from lipids and even from amino acids, glutamine and all that can be utilized to ultimately get the other important biomolecules. So, that is an important. Somewhere in 1979 P53 was identified and C-MYC also was discovered. These are all, people sometimes call this era as biochemistry era and genomic era. Genomic era is somewhere in 2000s, you see, when the human genome was ultimately deciphered. Of course, there is a theory that cancer is a genetic disease, mutated genes, they contribute to the transformation, and we have studied this, that is in colon cancers there are APC mutation. APC is a tumor suppressor gene, APC mutation, and adenomatous polyposis, RAS mutation, P53 mutation and several other chromosomal aberrations before you get a metastatic cancer. So, that we have studied, just this is recapitulating that. Now, somewhere in the genomic era, you also have the discovery that SRC and RAS regulate glycolysis, which means these are oncogenes. The oncogenes also contribute to the regulation of glycolysis. So, elevated levels of glucose transport and transporter messenger RNA are induced by RAS and SRC oncogenes. This was a publication. So, they are connecting, that is simply metabolic deregulation is recognized, but that metabolic deregulation may be contributed by a deregulation in oncogenes and tumor suppressor genes as well, and then hypoxia, all other parameters, ultimately signaling pathways also will be disturbed, that is there now. Finally, why we are studying cancer metabolism, an understanding of this, will it anyway help in treatment, forming treatment strategies. So, that is what we are going to end up the lecture with. Then c-MYC transactivation of LDH. LDH is that lactate dehydrogenase, implications for tumor metabolism and growth. People have started publishing that. So, mutated genes, that is oncogenes and tumor suppressor genes, also contribute to deregulated metabolism, and that may also help in the development of transformed cells. So, that is the idea. So, the control of metabolic switch in cancers by oncogenes and tumor suppressor genes. So, this is how, as the caption shows, cancer and metabolism came together. Again, people in the genomic era, again people have found mutations in mitochondrial proteins such as succinate dehydrogenase and the fumarate hydratase. We used to call it as fumarase, but some people call it as fumarate hydratase. So, these are enzymes involved in where? Which part of metabolism? Succinate dehydrogenase, TCA cycle, fumarate hydratase, both are in TCA cycle. So, you remember I told you Warburg said that there is some respiratory problem. So, accordingly in mitochondria also they found some problems. It is not that respiration is totally defective, but there are some mutations in mitochondrial genes. And c-MYC also regulates glycolysis, I already told you. P53 regulates metabolism, and people have also found that isolated dehydrogenase again in TCA cycle, there are mutations in this gene in the glioblastoma. Now we call this as metabolomics. You know what is metabolomics, right? The changes in the metabolites. You add omics with everything, proteome, microbiome, all kind of physiome even they say. The physiological changes are called physiome. So, you have so many omes there. Anyway, metabolomics is another, omics is also another thing, ome and omics are used. So, metabolomics is what people are talking about, and they have also found certain other enzymes like you see this PGHDH in is amplified in breast cancer, this is also a dehydrogenase. Then 2-HG, this is also a glutamate something, I forgot the full form, and isolated dehydrogenase, fumarate. The fumarate is also involved in induction of EMT. So, these are all things that connect the metabolism with cancer, that is what we are trying to say at various levels, various years from somewhere from 2000 to latest. That is why I said this is a very recent topic. So, cancer, could it be considered as a metabolic disease? This is a question. Now, the system view also has come. So, people have looked at the signaling cascades and genetic reprogramming, proliferation and all that, and finally, it is also connected with the nutrients and metabolic reprogramming like autophagy, many other phenomena could also be connected. If you take the history of this connection between metabolism and cancer, one of the important contributions is when the oxygen concentration goes down, that happens in cancer also, hypoxia. Then naturally you have this a conversion of pyruvate into lactate, as you can see here, glucose. So, the fermentative processes are accelerated when the oxygen goes down. When the oxygen goes up, right, now you have the TCA cycle and respiration. Pasteur effect is also connected with that, because what did Pasteur say? Pasteur's effect is slightly different. He said that oxygen will inhibit this process, glycolysis. That was his contribution. So, that is what he said, but it can be connected in this way, that is, if you go back, you see when the oxygen goes down, which also naturally happens in cancer because of hypoxic condition. That is how it could be connected with the Pasteur effect. Warburg experiments I have already told you. So, aerobic glycolysis in cancer cells, in the normal cell you will see that glucose, then under anaerobic conditions it will be converted to lactate, whereas in the cancer cells even under normal conditions you will have, normal oxygen concentrations, you will have the conversion from pyruvate to lactate. So, that is the important thing. So, this all came up that Warburg hypothesis being revisited, it came up as a big paper in 1956 in Science. The prime cause of cancer is the replacement of the respiration of oxygen by a fermentation of sugar. This is what Warburg said. He always thought that there is defect in respiration, which may not be totally correct. This hypoxic condition, even if it is not there, even if there is a normoxia, the lactate production will be there in cancer cells. So, that is an important point for you to just remember. This is what he proposed. Normal cell, there is injury of respiration, so that is phase 1, aerobic glycolysis phase 2, and de-differentiation, because cancer cells are poorly differentiated. The already differentiated cells will undergo de-differentiation and thereby they become cancer cells. This is what he proposed. This is the actual so called Warburg hypothesis. Just as there are many remote causes of plague, let us say heat, insects, rats, only one common cause like the bacillus phage, plague. There are many remote causes of cancer, coal tar, radiation, arsenic, pressure, so many causes, but there is only one common cause, the irreversible injuring of respiration. This is what he said, it need not be true. But then Weinhaus did not agree to that. He said there is no evidence that the respiration in cancer cells is either quantitatively lowered or fails to lower glycolysis. That is what he proposed, which is actually okay. Perhaps one idea is hypoxia, because perhaps tumor cells are glycolytic, but not inherently so, not aerobically glycolytic, but anaerobically glycolytic as a result of hypoxia driven Pasteur effect. That is what some people have said that. Warburg's experiment, the one problem was the Warburg's experiments were performed actually in well oxygenated tissues. That is why this confusion came in. Anyway, this is just to tell you about the historical thing. Now we have what is called the TCGA database. We can look at the various metabolites, the metabolomics comes into the picture, and see whether they are different from normal cells and cancer cells. What are the metabolites that are different? So, that kind of research is going on. The TCGA database, I hope you know what is TCGA, the cancer gene atlas database is there, and there are lot of other databases. So, from which what you can find differentially expressed metabolic genes. What you are going to look at is, you know what are the genes involved in various metabolic processes, you know the names, let us say 200, 300 or 400 whatever, right. You look at their expression in the normal cell and in the cancer cell, make a table and see which are differentially expressed. So, that is what people have looked, and then you look at what are the enriched metabolic pathways, what are the pathways that are going up or down in cancer in comparison to normal cells. People have done that, it is not that. So, this is what you get, metabolic landscape. Do not have to worry, they have looked at glycolysis, gluconeogenesis, oxidative phosphorylation. You see the oxidative phosphorylation is there already, it is not that it is not there, but it is relatively less. Purine biosynthesis, everything is there, upregulated, downregulated, all these things. You get something like a table like this. But now you can also look at survival of the patients, low survival, high survival, and then gene set enrichment analysis. This is how bioinformatics people look at it. That is what your metabolic pathways linked to patient survival, that is what they have tried. Now, this is the result. Here you can see oxidative phosphorylation is this blue color represents down, do not think it is up, it is down. Blue means enriched down, this color whatever you want to call, orange or whatever you want to say. What is the color you can say? Pink? What will you say? Whatever. the one that is not blue is enriched up. So, the blue color oxidative phosphorylation is down, whereas the purine biosynthesis and glycolysis, this is the most important point. Glycolysis goes up. So, that is what we have to know. That is related to survival. So, the point is oxidative phosphorylation is suppressed in low survival patients. Similarly, people have looked at tumors. They have seen that in some of the lung cancer patients, this lactate formation. Similarly, in renal carcinoma, lactate formation, those kind of things they have looked at. Mitochondrial dysfunction also was reported, like I told you already. Inhibition of oxidative metabolism leads to p53 genetic inactivation and transformation of neural stem cells. Stem cells are also affected. So, the basic point that you want to take home is, even under normal oxygen conditions, metabolism goes on, but oxidative phosphorylation is less. TCA cycle is not operated at a high rate, relatively at a lower rate, but glycolysis goes on at a higher rate, so that you will get more ATPs. But overall, this is an inefficient way of taking energy. Although it is inefficient, it is quick. That is why something like we say quick and dirty. That way the cancer cells utilize that. That is how it is. Mitochondrial dysfunction happens at different levels: succinate dehydrogenase, fumarate hydratase, and then isocitrate dehydrogenase. They are all mutated. So, this is an important metabolic dysregulation. I am giving you some examples. See, there is FH mutation, that is fumarate hydratase. I told you, in a disease called HLRCC, which means hereditary leiomyomatosis and renal cell carcinoma or cancer, whatever. It is an inherited disorder, increased risk of kidney cancer. It can also cause lesions in skin and uterus. So, this is a cancer where they have seen the germline mutations in the fumarate hydratase. They predispose to dominantly inherited uterine fibroid, skin, and the so called leiomyomata and papillary renal cell cancer. The main point that you have to take is that mitochondrial dysfunction is there, and that may be helping in developing cancer, certain types of cancer. That is all you have to take. That is the message. Fumarate accumulates in that what I said, that fumarate hydratase deficient cells. After all, this is the enzyme. If this enzyme is mutated, this one, this. If it is mutated, naturally what will accumulate? The fumarate will accumulate. The fumarate hydratase loss, I told you, if this enzyme is mutated, then EMT, even hypoxia inducing that inducible factor HIF, and then NRF2 are all activated. So, that may lead to not only cancer, but also to metastasis. Then there are some reports, reactivation of dihydroorotate dehydrogenase driven pyrimidine biosynthesis restores tumor growth of respiration deficient cancer cells. So, mitochondria, it is not that completely deactivated, they are also required. That is what is shown here. We have this is just explaining what I have just told you. Mitochondrial function is rewired in cancer cells, but mitochondria are required for cancer cell growth and proliferation. Mitochondrial dysfunction can activate oncogenic processes. That is what I just explained. Fumarate hydratase mutation can activate even EMT, NRF2, what is that, NR2, and what else I said? We looked at, just now yes, NRF2 and HIF, HIF is what I forgot, hypoxia inducible factor. Now, people have provided some evidence for the increased need of glucose by using this so called 18F, that is fluoro 2-deoxyglucose. what is called FDG, positron emission tomography. By this imaging or some kind of analysis, you can actually look at this is how the machine looks like. This is a PET instrument you must have heard about. So, it shows increase in glucose uptake in cancer. So, this is one of the proofs. If you give some radioactive glucose, it accumulates. So, they see that there are a lot of, I mean, it is taken up by many cancer cells specifically. So, that is what they have shown, and they have proven this by several other imaging methods. That is, some of the metabolites have been radioactively labeled, and they have found the accumulation of this radioactivity in those metabolites. Like here you can see MRI, they have used C13 glucose, C13 urea, C13 lactate. By all this so called magnetic resonance MRI, magnetic resonance imaging, they have shown the tumor glycolysis using C13 labeled glucose. So, this also proves the point that cancer cells indeed need more glucose. So, this is also same thing, just another figure, that is all. So, what happens, why there is an aerobic glycolysis, we say that it is increased, but it is a combined result of defects in oncogenes, because the oncogenes are getting activated, proto-oncogenes are converted to oncogenes, and tumor suppressors are deleted or inactivated. There is a hypoxic microenvironment, there is also mitochondrial DNA mutation. So, all these factors contribute to increased aerobic glycolysis. That is what it is. Is it all about it? This is just a cartoon, do not worry. So, the aerobic glycolysis also supports proliferation, because after all you need energy for proliferation. People have done several kind of computational studies. Migration also, computational study of the Warburg effect identifies metabolic targets inhibiting cancer migration. So, let us look at it, this is somewhat important. LDH upregulation, LDH is lactate dehydrogenase. LDH, this is important for hyperplasia to neoplasia. LDH expression drives tumor formation in the context of EGFR overexpression. Yeah, this is also another thing. In this manner, EGFR overexpression automatically it leads to proliferation. So, that is there. Increased sugar uptake is also in Drosophila EGFR model, they have shown this. Synergy between EGFR and LDH correlates with poor outcome in human cancer. So, there are what we are trying to say here is lot of experimental proofs have shown that overexpression of glycolytic enzymes may help in cancer cell proliferation. So, that is the idea. So, aerobic glycolysis is a hallmark of cancer. Aerobic glycolysis is a characteristic feature of most proliferating cells. Aerobic glycolysis provides building blocks for cancer cell growth. So, these are all what we understood from whatever we have said now. This is again Warburg hypothesis is not necessary. It is also involved in stem cell proliferation. Stem cells also may undergo a similar way, they may also alter glycolysis. As you can see here, when the glycolysis is up, you see the stem cell renewal, self renewal. They can also go into differentiated cells. So, there is a inverse relationship, glycolysis is up, oxidative phosphorylation is low, that is what is shown here. Then finally, the question is whether we can use this, exploit this altered cancer metabolism for therapeutic strategy. That is the most important question. After all, we are studying all these things. We saw how many people have provided different kinds of evidences for altered metabolism in cancer cells. and in different processes, mitochondria, oncogenes, tumor suppressor genes and so on. So, can we exploit this improved knowledge on cancer metabolism for designing certain therapeutic strategies? That is the question. The answer is yes. The first example of targeting cancer metabolism, perhaps you know, the best journal in the medical field is this New England Journal of Medicine. If something comes in this, then it is talked all over the world. So, this is that is why they are giving this example. Temporary remissions in acute leukemia in children produced by folic acid antagonist 4-aminoterryl glutamic acid, aminoterin. The commercial name is aminoterin. So, this is one of the examples, because in 1948 itself people have shown that aminoterin could be used as a therapeutic agent. So, people have used several from since then lot of other molecules have been found. Basically, we said glycolysis is up in cancer. So, if you prevent that, the energy consumption will go down. So, the growth will come down. That is the whole idea. So, you need to find molecules which will inhibit glycolysis. So, that is what is shown here, some of the examples: fluoritine, methazolamide, indicylphone. You see the reaction which is affected by these chemicals. For example, what is this enzyme? Anybody knows? Hexokinase. So, that is lonidamine, 2-deoxyglucose, bromopyruvate and so on. Similarly, here the nucleotide synthesis can be inhibited by 5-fluorouracil. These are all well known anti-cancer agents: gemcitabine, clofarabine, mercaptopurine, methotrexate. These are all very well known. They act, this is you know this pathway of nucleotide synthesis. Anybody knows what is it called? Nucleotide metabolism, salvage pathways and all, you have studied? Yes. So, they act on that. Again, glutamine is another important component. So, phenylacetate, it will prevent this glutamine related pathway. Similarly, what else? For fatty acids also you have certain chemicals, and what is other important thing here? Yeah, lactate, again you have certain chemicals. So, these are some, yeah, isositate dehydrogenase. This is also inhibited by some other chemical. That is okay, it is actually a lot of cancer anabolic metabolism inhibitors actually move into the clinic. So, they have given some examples. I have given a table, you can look at it. One is highlighted for renal cancer and breast cancer, phase 1 and phase 2 trials. The glutaminase is the target. Then lot of isositate dehydrogenase, lot of such arginase. Several companies are interested. You can see the company names here. The targets you can see here, most of them are metabolic enzymes. What is the target? The enzymes in related to glycolysis or the other processes that I mentioned. So, they are the targets. So, it is a very active field of research. You can see some of them have gone to phase 3 trials. So, that means we are almost nearing completion of that. So, what is the conclusion? Metabolism of cancer cells is different from that of normal cells. Dysregulated metabolism can drive oncogenic process. Altered metabolism offers a therapeutic window to target cancer. Of course, I took all this from internet, but this appears to be a good one. The references also they have given, you can look at it. We are done. So, we have kind of completed. This is one thing that I have not taught before, because it is only a kind of recent topic. So, with this we are coming to an end. So, we will meet only in end semester. So, of course, the WhatsApp group is there. If you have any doubts or something, you can always ask. And I will give you, I have given you some wonderful reviews. Even there is a PDF file, you can look at it. because these slides won't tell you the actual story. That's why I gave you the PDF. You can read that. It's a very interesting write-up. Actually, one of the best reviews only. There are many reviews, but I have selected some, especially the EMT one. That itself will give you an idea of cancer metabolism also. That also, it's written by Sendurai Mani, is a friend of mine. He used to be at MD Anderson. Now I think he moved to some other place. He is very good at that. He is a postdoc of Bob Weinberg also, and he has really given the latest developments on the therapeutic side. What is the knowledge gained and what are the therapeutic avenues, including cancer cell metabolism. Because the topic is EMT, but he has also touched upon cancer metabolism. So, you can look at it. I think if you really want to know more, these references should be able to help you. And you can look at some of our publications also. If you want to gain some knowledge on microRNAs, we have recently also we have published. So that you will have some idea of how people are working on these areas. Of course, we cannot cover everything. I hope I have covered most of the important topics.